Alright guys, in this little screencast, let's talk about DNA and let's talk about how we can separate it and move it and organize it. And what the difference between this like chromatin and sister chromatids and homologous chromosomes and all that other stuff is. And let's try and understand this really quick. So I want you to look at this first picture right here and imagine this is your nucleus or inside of your nucleus. So I could take a marker here and I could draw a circle and be like, here's my nucleus. So when you were learning about cells, when we were learning about cells earlier, we labeled all this stuff in here the chroma. Oh man, I hate this pen. Here we go. We labeled all this stuff the chromatin, right? So here's our chromatin. Now, this is all DNA, all spread out and untangled. And if I were to say to you, like, okay, sort that for me. Pick up the red and put it over here, and pick up the blue and put it over here, and pick up the white and put it over there, it would take hours. You know from life experience that that's going to be all knotted and tangled and just an absolute mess and nightmare to sort. However, this wouldn't be so bad. You could sort this red and purple and pink. You could just pick them up and move them, no problem at all. The problem is DNA can't always be like this. It has to be like this. Because in order to do all those things we've been talking about, replication, where we make 2 copies of the DNA, or transcription, where we write a message of mRNA to send out of the nucleus out to the cytoplasm, to the ribosome, DNA has to be able to unzip. So it can't be all coiled up and stored nice and neat like this. So how do we get from here to here? And why is it necessary? Well, here's what happens. Here we have our chromatin. And they're showing you how the DNA starts to wind around all these, I guess, nucleosomes and histones. So we see a DNA helix here and how it's wound kind of around that stuff. And eventually it forms this condensed chromosome. Now, it might be a little easier if let's do this. Let's get rid of this guy over here, make him a little bigger. It might be a little bit easier if we kind of just break it down. So here we have our gene, we have our DNA, it's unwound. We can unzip this, right? We can bring in some helicases, unzip it, we can replicate it, we can make mRNA and whatever. But over here we have these proteins. And now we're starting to wind the DNA around those proteins. Then we take those proteins and kind of wound them around themselves. And then we wind it some more. And eventually we get a chromosome. So the whole point of this is, during the cell, when the cell is growing and doing what the cell needs to be doing, and the cell is replicating, so we're talking about that G phase and that S phase, that G1 phase and S phase of interphase, the chromosomes, we don't see them because they're all unwound. But when we get to the phase where we need to start dividing, right before prophase, we need to wind all these guys up. So here I have 2 pictures of a cell, and you know that this is a cell in interphase, and you know that this is a cell in prophase. And why I put them in is just to show you, hey, look, right here is all my chromosomes, my chromatin, so it's all this DNA unwound, and then when we go over to prophase, now we see these little wound up bundles. So I want you to think back to that first slide. It's just like, here's my spool of thread, all unwound, and then when it gets time to sort everything out, we wind all that string up on a spool of thread. We wind all that DNA up into a chromosome, and that's all this is. But there's different types now that we've said that, there's other words that you've been seeing. You've probably been seeing chromosome, homologous chromosome, sister chromatid. What is all that stuff? So let's take a look and see if we can't figure that out. Here is an unduplicated chromosome. Okay, so this is a chromosome. Now we wouldn't have it like this before we duplicate it because it would be all unwound, because in order for replication to happen, it has to be all unwound. But there is one point in the cell where it looks like this. We'll talk about that later. So think about this now. I have 2 of those unduplicated chromosomes together, and they have it labeled as duplicated, aka replicated chromosome. So this are my 2 copies of DNA. These are my 2 copies of DNA. Now remember, DNA, when it replicates, is semi-conservative, so half of this chromosome is the original piece of DNA and half of that chromosome is the original piece of DNA. And then over here they say we have a pair of duplicated homologous chromosomes. Now, I'm a firm believer, if you get what I have right here, this will make everything in your life so much easier when we talk about mitosis and meiosis. How do we get it? Well, guys, you need to have in every cell a chromosome from mom and a chromosome from dad. So mom gives you this chromosome, dad gives you this chromosome. They're the same. In other words, say right here was a gene for eye color on mom's. Well, it would be here too, because these two pieces of DNA, this piece of DNA and this piece of DNA are exactly the same. They're replicated from each other. But over on dad's chromosome, you might have a gene for eye color right here, and you might have a gene for eye color right there. Because again, these are replicated, so semi-conservative. These two pieces of DNA are exactly the same. But this chromosome came from mom, this one came from dad. And maybe mom gave you a gene for brown eyes, and dad gave you a gene for blue eyes. So mom's chromosome that she gave you has eye color on it. Dad's chromosome that he gave you has eye color on it, but the gene for eye color might be different. Like I said, mom might have gave you brown, dad might have gave you blue. So what we have, and you got to get this, and if you don't get it, come talk to me. What we have are two homologous chromosomes. One that came from mom, one that came from dad. They carry the same gene. The trait might be different. The actual phenotype, what it looks like, might be different. So what we have in describing all these things, just to recap, we have a chromosome, and then this right here, and this right here we call sister, I didn't tell you this before, chromatids. So those are sister chromatids because they're exactly the same, they're replicated. So I have a sister chromatid here, and a sister chromatid here, and I have a sister chromatid here, and a sister chromatid here. And then somewhere else in the cell is another chromosome just like this one that came from the other parent that carries the same genes but maybe different traits. One last thing. These are connected by these big dots we see, right? Those dots are called centromeres. Down here it says centromere. And on each centromere there's something called a kinetochore. The kinetochore is what allows those spindle fibers you've been reading about to come in and grab the chromosomes and start to pull them apart. All right, hopefully that makes some sense. And if you have any questions, certainly come and see me.